SISSGEO is a partnership between the Brazilian Health Institute Fiocruz and the National Laboratory for Scientific Computing. It was released in March of 2014 and is one of the Brazilian actions to achievement of the Aichi goals by 2020. Its development is part of a national project for biodiversity endorsed by the GBIF. Health impacts by biodiversity loss and monitoring of pathogenic agents on nature and on the borders between rural-urban environments are challenges for countries with continental dimensions and great biodiversity. Many impacts drive the ecological opportunities for the emergence of pathogens. The major are advancement of agriculture and livestock, changes in terrestrial and aquatic ecosystems, deforestation and reforestation, climate change, human density on the banks of natural areas, changing the new Brazilian Forest Code 2012. Health surveillance in Brazil is divided into human and livestock sectors, and the zoonoses arising from wildlife are considered only classical endemic diseases or emergencies. But in many places, especially in the remote ones, humans and animals are in close contact, like the wildlife cycle of yellow fever. As the connection between primates and humans in semi-arid regions of the Caatinga of Brazil, there is no program to monitor wildlife health and the potential spillover of parasites to humans or livestock. The problem is not new. But in the last years, the emergence of influenza virus and now the Ebola show us how important the impact of the biodiversity loss to health. For example, mining and other causes of deforestation in Brazilian Amazonia destructures the forest, contributes to the loss of primate species and exposes workers to the malaria's mosquitoes vectors. So, as we can see, the malaria is clearly an environmental problem that in a complex life cycle involves a lot of plasmodium species, mosquitoes and primates, that in a natural stage plays a delusion effect that is broken by the humans. So, the challenges to monitor wildlife in mega-diverse countries, especially in Brazil, are continental dimensions, parasite vectors and hosts diversity, lacks of biodiversity knowledge, high complexity of the disease's ecology, methodology and infrastructure bottleneck for big data, socio-cultural diversity, human health emergency, change on epidemiology profile from social drivers. In order to consolidate available knowledge and to create agile solutions for monitoring and preventing wildlife diseases, this year the Oswaldo Cruz Foundation developed the Wildlife Health Information Center, a virtual space whose the central focus is the Wildlife Health Information System, SISSGEO. This center aims to consolidate and disseminate the knowledge of relations between wildlife and human health and inspire society participation to monitoring wildlife health and improve its quality of life and conservation. The Wildlife Health Information System, SISSGEO, is the central focus of the center. SISSGEO is one of the data sources for the Brazilian Biodiversity Information System in conjunction with the biological collections of all Fiocruz. The data supply to GBIF is part of the institutional and the Brazilian government policy that seeks to integrate national and international databases. Our general architecture includes application, database, and high-performance servers. Our system is connected to geographical and biological databases, such as GBIF and the Brazilian Biodiversity Information System, SIBBR. It collects data of animals and their physical conditions in sight. All citizens, researchers, and health, environmental, and agriculture agents are stimulated to participate. The data collected are processed through mathematical models considering hierarchical and environmental parameters, generating alerts and wildlife disease prediction. When the mathematical model indicates an alert, the government services are notified and biological samples may be collected. With the reliable diagnosis improved by the Wildlife Health Laboratories Network, the models can be validated and so train the model that can be used to construct the prediction models in the future. The workflow of the analysis can be described as: a new register is added to SISSGEO. An unsupervised learning technique defines if the register belongs to a previous event or group or if it will form a new event. A feature extraction technique is applied to describe the event. Supervised learning is applied to classify the event as an alert or not. If the event is classified as an alert, the information is sent to a network of laboratories and the Wildlife Health Diagnosis Network, which will then confirm whether the event is actually an alert, validating the classification process and augmenting the training data set. Our tests with the proposed mathematical modeling show that the majority of intestinal health means diversity in an endemic rodent living in the semi-arid region of northeastern Brazil is most distributed in areas with altitude between 438.8 and 888.1 meters. Considering the model, these areas are those where potentially the transmission of parasite may be higher. Other models developed for mammals indicate the same region as most important for conservation in semi-arid Brazil. Both results suggest important questions. 1. The diversity of parasite is an important component of the evolution of species biodiversity and should be considered in conservation projects and demarcation of protected areas. 2. The parasite study can be cheaper and easier than the study of larger species, especially if indirect research methods are used, as proposed. 3. The semi-arid Brazil has less than 1% of its preserved area, and this result confirms the need to deploy new conservation areas. 4. Knowing the parasitic transmission potential, both the decision-makers of the human health system and the wildlife protection may establish policies and action for disease control and prevention. Comparing the data used in the experiments carried out in northeastern Brazil with rodent data recorded and available on GBIF, we can consider some collaboration scenarios between SISSGEO and GBIF. The first is the validation of the contribution of the data included in the GBIF by SISSGEO team. In the study, we identify taxonomic misidentification of species or georeferencing in 2 records. The second is the use of GBIF data by SISSGEO for taxonomic validation of volunteers' records, when they are faced with the distribution areas of validated species. Then, the use of GBIF data for modeling the parasitic transmission regions by overlapping host and parasite niche distribution. Finally, the availability of SISSGEO data included on the Brazilian Biodiversity Information System SIBBR, which is the Brazilian GBIF node. Until now, we have 70 wildlife and parasitologist specialists, 49 laboratories in the networks, and about 1,400 people in Facebook. To make sure that common people will be able to use the system, we work together with the network specialists and field workers too. Define data required, simplify screen layout, reduce amount of information and controlled fields, simplify taxonomic groups, symptoms, and physical aspects related to the potential for transmission of pathogens to widely familiar words and concepts. Differentiate the spatial visualization of users' data from other users. Implement the automatic georeferencing even without a connection of internet. Implement the ability to send photos and videos. The growing use of information technology and mobile communication reach more than 50% of the Brazilian population, and this opened up the possibility for people from the most remote locations to participate in SISSGEO. Now, the app is available at Google Play and the Apple Store. The SISSGEO is 100% free. The results provided by SISSGEO are available and associated with disease prevention information and good practices for health and biodiversity conservation in SIS website. Now, the important challenge is to mobilize citizens and specialists to use the system. We thank the team that is building this idea and the Global Environmental Facility that sponsors this project, with the World Bank and the 8 institutions that with us are part of the Brazilian project to biodiversity, ProBio2.